Welcome everyone to another exciting edition of NeuroConnect, our weekly live event dedicated to unraveling the wonders of neuroscience. Together with eminent experts in the field, we aim to support and empower both patients and caregivers as they navigate the challenges of living with neurological conditions. Our focus this week is on the prevention of Alzheimer's disease. Alzheimer's disease, a condition that impacts millions of lives worldwide, has brought our attention to the importance of memory and cognitive health. Throughout this event, we will delve into the latest research, insights, and strategies that can help us better understand Alzheimer's disease and its prevention. Our aim is to equip you with valuable knowledge that can empower patients, loved ones, and the community to take proactive steps towards maintaining optimal brain health. In today's event, we will first discuss a recent finding, the potential use of a finger prick test for the early detection of Alzheimer's disease. Then we will hear from our distinguished guest, Dr. Vishwambar Bhandare, who will be sharing his expertise and prospects on the crucial subject, from understanding the science behind Alzheimer's to exploring lifestyle factors that play a role in prevention. We have a wealth of information to uncover. Before we begin today's session, we encourage our audience to ask questions and share experiences as we strive to make this event interactive and informative. We will be addressing these questions at the end of the session. Let's begin today's session by understanding a research study that investigates the possibility of using a finger prick test to identify Alzheimer's disease at an early stage. As per the study findings presented at the Alzheimer's Association International Conference in Amsterdam, Netherlands, researchers found that finger prick test may be effective for diagnosing and monitoring Alzheimer's disease remotely. As per another study, researchers found that blood tests deliver over 85% accuracy in Alzheimer's diagnosis, whereas primary care physicians have around 55% accuracy. This advancement has the potential to enhance both the precision and accessibility of Alzheimer's disease diagnosis and tracking, offering a promising avenue for early intervention and delaying the onset of the disease. As we know, currently there is no cure for Alzheimer's. However, studies show that early diagnosis and intervention are crucial for delaying its onset. While current diagnostic methods involve MRI, cognitive assessments, and physical examinations, their accessibility remains limited. These techniques require visits to specialized clinics staffed by trained personnel and involve complex procedures for sample transportation and storage. Further, the accuracy of these methods varies, with autopsy results revealing that around 25% of clinically diagnosed Alzheimer's patients did not exhibit conclusive evidence of the disease. Moreover, a significant proportion, up to 50% of individuals with dementia, are not formally diagnosed during their lifetime. The emergence of at-home blood test for Alzheimer's presents a promising breakthrough. A study conducted in Barcelona, Spain, included 77 memory clinic patients who provided both venous and finger prick blood samples as well as neuropsychological measures. These samples were analyzed for Alzheimer's related biomarkers, and the results demonstrated the potential feasibility of using finger prick collection and dry blood spot cards for quantifying biomarkers and monitoring neurological conditions. Another study from Lund University, Sweden, highlighted the diagnostic potential of blood-based biomarkers achieving an accuracy of 85% compared to primary care physician examinations. The imminent availability of this blood test could transform Alzheimer's diagnosis and treatment strategies, leading to an era of enhanced diagnostic accuracy and improved patient care. To drive this further and strengthen our understanding, today we are privileged to have Dr. Vishwambar Bhandare. Please join me in welcoming Dr. Bhandare, who is working with Innoplexus as Associate Scientific Manager. He brings with him a wealth of knowledge, expertise, and a deep passion for research. As a testament to his contributions, Dr. Bhandare has authored over 25 research articles published in national and international peer reviewed journals. Welcome, Dr. Bhandare.
Thank you, Deepa.
Thank you. Thank you for joining us. Over to you, Doctor.
Yeah, thank you. So, I hope screen is visible, right?
Yeah, your screen is visible.
Sure. So, today's title for this Neural Live event is Memory Matters, a deep dive into the Alzheimer's prevention. So, question here is, does memory matters? If you see in this slide, I have shown two characters. One is male character, in the another is female character. So they try to recall the things that happens before 5 years. If you see the male character, he is not able to recall the things before 5 years clearly. And if you see the female character, the lady is able to recall the things what happened before 5 years perfectly, right? So if you see which one is the better in our understanding, so if we recall all the things, the good memories what we have, that is good, right? So I would like to give some examples here. So if what will happen if our memory has a problem? See, here, if it would be very difficult to recognize family members and relatives, very difficult to interact with friends. We face problems with direction and maps to go out. Office work and daily meetings would be difficult if we have a memory problem. Most importantly, ATM pin we cannot remember, and very difficult to get withdraw money whenever we need. Also, other financial transactions you would not be able to do using any UPI apps or net banking credentials, as we are not able to remember this login details, right? So these details we cannot get anywhere. So this we should remember by ourselves, right? So if you see all these cases, this memory, we get the answer. Yes, memory really matters a lot in our life, right? Now, I would like to highlight the connection between Alzheimer's and memory. So, if we see this slide here, we have discussed some points. I would like to highlight when we have a confusion with time and location, we have difficulties in completing familiar tasks, we have we have difficulties in problem solving skills, problem with memory loss, misplacing items, poor judgment, difficulties with the words, trouble with images and spaces, and difficulties feels like withdrawal from the societies, then we can suspect the possibility of Alzheimer disease. Right? In general, Alzheimer disease is most common cause of dementia that triggers a gradual decline in memory, thinking, and behavior and social skills. So this memory is very important for all of us in our life. Right? So this is the overview of various hypotheses reported in Alzheimer disease. I have summarized few which are most popularly discussed. Here in the first panel, I have shown picture of healthy brain and Alzheimer disease brain. And if we see the pictures carefully, we see the size of healthy brain is relatively larger when we compared with Alzheimer disease brain. In Alzheimer, what happens? It causes the brains to shrink as the disease progresses, right? And panel B represents the classical hypothesis of Alzheimer disease, that is amyloid beta plaques and neurofibrillary tangles. Now we see the Alzheimer brain picture at the corner, and we see structure of neuronal cell. We can see the brown patches. These are the amyloid plaques, and in the red color we have shown neurofibrillary tangles. And in this interesting thing is like this, below we have shown the red color tau protein, which is bound to the microtubule. When it is hyperphosphorylated, it detaches from the microtubule and forms aggregate. Now, interesting thing is this microtubule is made up of alpha beta subunits, right? And heterodimeric subunits. And interestingly, it is now well studied that the beta and alpha isotypes, they have multiple isotypes, and their expression pattern varies greatly. And in cancer also, their expression, differential expression has been reported, and also in neuronal cells and various neurodegenerative diseases, this isotypes expression varies. Now, if you see the in each subunits, for example, if we take beta subunit isoforms, beta subunit plays major role in cell viability. Beta 2 isoform is responsible for neuroid growth, outgrowth. Beta 3 is helps neuronal cells against free radical and reactive oxygen species. Surprisingly, expression pattern varies greatly, and there is no specific ratio has been studied. However, we notice differential mixed population of alpha and beta subunits. In addition to this, if we see panel D, there is a direct connection between microbes and Alzheimer disease. The microbes have direct and indirect association with the Alzheimer's, and the relation is well established using oral gut-brain axis. Similarly, various viruses, including herpes virus, cytomegalovirus, and HHV6 virus, there are direct evidence that they are responsible to trigger the Alzheimer's in many cases. So many other hypotheses are also being reported for Alzheimer's. Now, if we see the disease progression, it starts with the preclinical state. Preclinical state means we don't see any significant symptoms. However, brain undergoes changes in the biological function. If it progresses to the mild form, followed by moderate and severe Alzheimer disease. If you see the location, the picture below represents the brain part which is affected starting from the mild, moderate, and severe dementia. If you see carefully, in the mild form, uh, we see medial temporal lobe is affected, which progresses, which further affects to the lateral temporal lobe and parietal lobe, and which spreads further to the frontal lobe, and followed by occipital lobe, which similarly slowly it progresses across the brain, which is causing severe form of dementia. So overall, if you see, the progression is slow, and we see more visible symptoms that affects our daily life activities. Now, question is why Alzheimer matters. It is very important to note neurodegenerative we have many neurodegenerative diseases, of which Alzheimer is leading to dementia, and it is worldwide around about 55 million cases has been reported so far, and number is increasing. Early detection of Alzheimer is difficult. We have limited treatment options available to treat, and if you see, good thing is that the number of clinical trials uh, being used are more, and now 1,000 trials are in progress. If you see the grants, research grants, 7 grants are awarded till date, and the value worth $2.3 million. So this data we have collected from the OntoSite. Now we'll talk about risk factors for Alzheimer disease. In Alzheimer, risk factor. Before I would like to, before discussing the factors, I would like to highlight what are the risk factors. In risk factors, these increase the chance of getting disease. However, they are not cause of the disease, and increase the risk factor increases the chance but not certainty, right? So there are two broad categories in the risk factors. One is modifiable risk factors and non modifiable risk factors. In modifiable risk factors, we have high blood pressure, smoking, and in non modifiable we have age, genetic makeup. So these are not the only factors we have. To summarize, I have highlighted few. In detail, I will explain all the possible factors in upcoming slides. However, it is very important to note having little or no exposure to the known risk factor does not necessarily protect a person from developing the disease. Now, I would like to go focus on modifiable risk factors in the Alzheimer. Sorry, people, if you see the pictures, I have highlighted high blood pressure, smoking, diabetes, cholesterol, obesity, poor diet, and various other risk factors. I will explain one by one. People who have high blood pressure, that is hypertension, are more likely to develop Alzheimer disease when compared to people having normal blood pressure. Since high blood pressure affects the heart, arteries, and blood circulation, it can increase the risk of developing Alzheimer disease. How we can lower the blood pressure? Research shows that physical activity and healthy diet can lower the high blood pressure and reduce the risk of developing Alzheimer. Another factor is cigarette smoking is related to, as we all know, cigarette smoking is related to wide range of disease including many forms of cancer, cardiovascular disease, and diabetes. The evidence is strong and consistent that smokers are at 45% higher risk of developing Alzheimer disease when we compare with non-smoker or ex-smoker. It is never too late to quit. Smokers who quit the smoking reduce their risk of developing dementia. Now, people who are suffering from type 2 diabetes are at the midlife age. They are also having higher risk of Alzheimer disease. How we can control the diabetes? Careful management, such as regular monitoring of glucose level, exercise, which they can reduce risk of developing cardiovascular disease and dementia, right? Next, people with high cholesterol level in the midlife are also likely to develop Alzheimer disease when compared to the people having normal cholesterol level. High cholesterol level also increases the risk of hypertension and diabetes. Further, obesity and lack of physical activities also contributes to the disease development. Primarily, diabetes and high blood pressures are the more easily prone risk factors for obesity. as well as obesity in the midlife increases the risk of developing Alzheimer disease. Interestingly, unhealthy diet, including high saturated fat, sugar, salt, can increase the risk of developing many illnesses, including type 2 diabetes, cardiovascular disease, and Alzheimer disease. The healthy diet can reduce the risk of developing dementia. The healthy diet includes Mediterranean diets and DASH. DASH represents dietary approaches to stop hypertension. So both the diets recommends low intake of meat, sweet, dairy, and a high intake of fruits, vegetable, legumes, whole grains, nuts, olive oil, and fish. So if we change the healthy lifestyle, healthy diet, it will reduce the risk of developing Alzheimer disease. Another factor is alcohol. Is the fifth, as we all know, alcohol is the fifth amongst most important risk factor for death and disability worldwide. Low to moderate level of drinking may reduce risk. However, most specialists do not recommend this as a way to reduce the risk factor of dementia. People who actively use their brains throughout their life may be protected against brain cell damage caused by Alzheimer disease. Now, people having depression or brain injury, hearing loss, are also having higher risk of developing the disease. Social isolation can also increase the risk of hypertension, coronary artery disease, depression, and dementia. So, how we can monitor, regulate this? Staying socially active may reduce the risk of dementia. Social interaction may also help to slow down the progression of the disease. Another important risk factor is air pollution, living near busy roads or highways. So it is estimated that people who lives within 50 meters of busy road or highways are more likely to develop dementia. This is mainly due to the hazardous air pollutants which are coming out of vehicle smoke. Right? So these all are very important risk factors in Alzheimer. So there is no cure for Alzheimer disease yet. However, certain strategies, lifestyle changes would help to reduce the risk of Alzheimer disease. Sorry. The slide represent some of the recent articles which are highlighting these risk factors which we have now discussed in the last slide. If we see the carefully, exposure to traffic generated pollutant is also responsible to affect pathophysiology in Alzheimer disease. If there is a direct connection well established between alcohol use and Alzheimer disease progression. It is interesting to note ultra processed food consumption is increases the risk of dementia. So ultra processed food means the food, it is frozen food what we eat in McDonald's, Burger King, right? And traffic related incidences are also proven their role in the Alzheimer. If we see type 2 diabetes and Alzheimer has very close relation. If we decrease the alcohol consumption, risk of dementia is also decreased. And if we see, depression is also directly associated with the Alzheimer, and people with hearing loss are also having close association with the disease. In the non-modifiable risk factors, there are people having age of 65 are more having the risk having double the risk of developing Alzheimer's. There are many genes which are reported in Alzheimer disease. There are more than 20 genes in which several mutations are reported that causes the Alzheimer. And gender is also responsible for the Alzheimer disease as a risk factor, because women are at high risk of developing Alzheimer disease than men. The reason is unclear. However, there are many research, they suggest the estrogen level of women varies across the lifetime, and that would be some responsible factor. Now, these are some examples which talks about, these are the examples for non-modifiable risk factors. Now, I would like to highlight the therapeutic approaches briefly. The drug-based approaches, the treatments are there, and non-drug-based. Drug-based approaches helps to improve the symptoms, and some drugs helps to modify the disease biology. If you see in drug, small based molecules are there which are used to treat the or relieve the symptoms, and monoclonal based are also there. And there are in non-based, there are therapies like physical activities, memory improvement, and all. Now, several clinical trials are undergoing for Alzheimer disease. We hope to get better and more efficient drugs soon. How we can reduce the risk of developing Alzheimer disease? Be physically active, avoid smoking and excessive alcohol consumption, track your numbers. The number is blood pressure, cholesterol level, blood sugar, weight within the recommended range. Stay socially active, interact regularly with others, make healthy food choice, eat balanced diet, reduce stress, challenge your brain by trying something new, playing games and learning new languages. Protect your head by wearing helmet when we engage in sports activities. Consider installing handrails on stairs and grab bars. These are, now I would like to conclude. 90% of us can avoid ever getting Alzheimer disease if we do some lifestyle changes. However, 10% people who are having more risk of genetic factor for cognitive decline, they can potentially delay the progression, we can slow down the progression of Alzheimer disease. Thank you from my side.
Thank you, Dr. Bhandare, for this enlightening presentation. I'm sure this session has greatly educated our audience on the significance of Alzheimer's prevention. Now, let's take questions from the audience. There are a few questions. So, the first question is what is the difference between dementia and Alzheimer's?
So, dementia is a general term for a decline in memory ability, which severely affects our daily life, while Alzheimer is a specific disease which slowly progresses.
Okay, thank you. There is another question wherein the user wants to know what is the average life of an Alzheimer's patient.
On an average, person with Alzheimer lives 4 to 8 years after diagnosis, but they can live as long as 20 years. However, the brain shows some changes before diagnosis of Alzheimer disease, but definitely our lifestyle change would help us to increase the disease progression further.
Okay. Um, any novel drug that has shown effectiveness or effective outcomes in the undergoing clinical trials?
Yes, recently tau targeting antisense gene therapy. The tau targeting antisense oligonucleotide has shown great success in phase 1B of clinical trial, which helps to treat mild Alzheimer disease. One more example, successful example is lecanemab. So that shows great result in phase 3, and that is approved by US FDA for early Alzheimer disease treatment. Similarly, many antibodies and small RNA-based therapeutics are undergoing clinical trials. Hopefully, we will get good molecule or drugs soon.
Yeah, thank you. Thank you, doctor, for addressing these important questions and providing valuable information about Alzheimer's prevention. Thank you for joining our event today.
Sorry.
Hope to have you with us again in the future as we continue to raise awareness about neurological disorders.
Sure.
Thank you.
Thank you, Deepa. I would also love to contribute my expert. Thank you.
Thank you. As we conclude this event, we encourage you to continue seeking information, staying connected with relevant organizations, and prioritizing brain health in your everyday life. Together, we can make a positive difference in the fight against Alzheimer's. Now, as we have come to the end of the session, I thank you all for being so engaging. Join us again next week as we explore empowering youth, understanding migraines in young minds with Dr. Surendra Koshia. Until then, take care and stay curious. Thank you.